Hey, Kiara.
Yep,
How's it going?
it's good. How are you
It's
doing?
it's good. I am swimming.
Good.
I just did the first session of live coding in my applied stats class, and it was not what I would call smooth or seamless, but we managed to survive
There's
barely.
always next time, right?
What's that?
I said there's always next time.
Yeah, it gets easier as they get more comfortable with it. And on Monday, I think we're going to do it in person rather than on Zoom. So that should hopefully make life easier.
Yeah.
Unfortunately, there was a problem at the end, and so I'm getting to class a little bit late. And as a result, I also didn't upload something to Moodle that I wanted to upload. So let me do that real quick so that you have access to today's PowerPoints. Hopefully I'll be a little more prepared for next week. How was lab yesterday?
I thought it was good.
And so how do you guys feel about handling the specimens and all that jazz?
It got better as I did more. At first, I was
Oh,
really
did it?
grossed
Okay.
out. Then I got more
All right.
comfortable with it.
Okay, good. I can see why it's a little bit weird at first. Oh yeah, okay. I'm having to remember where I put something. And unfortunately, I have too many things plugged in, and really I don't have enough ports on this computer. Unfortunately. All right. Screens. Where did you guys go? There are you. All right. So I need to just go and find the right file. Okay. If you go to Moodle, there's a 22nd of January 2021 folder, and in there is the PowerPoint that I'll be running through today. Okay, can you guys see my PowerPoint?
Yep.
Okay. So, well, before I do this, one of the things I want to do today is I want to talk about the history of herpetology. One of the things that I think we don't do a great job of in the biology department is I don't think we do a very good job of talking about the history of the field of biology. I don't know, how do you guys feel about us doing a good job of that or not?
I feel like maybe we did an E&E, but that was a long time ago.
Yeah, so I think it's important that we kind of understand sort of where the ideas that we currently teach you came from. And I think we sometimes get so caught up in working on getting you to learn what the ideas are that we don't give you a sense of kind of the progress of science and how science has changed over time. And that's something that I try and address in the classes that I teach, which is probably why the only time you remember getting much of it is back in evolution and ecology. So what I want to do is I want to go through a brief outline of the history of herpetology, so that you have kind of a historical context for why we think about certain things the way we think about them. But before I do that, let's go through the syllabus. So there is a syllabus on the Moodle site. And so you can either go and download that yourself, or you can just follow along with me. But we didn't start with that on Wednesday because I kind of wanted to go through the material on Wednesday in an effort to kind of get you to buy into the class, since all of you are just seniors taking this as an animal class. So my computer's running a little slow because it's compiling the video from the class that I just got out of, I think. So everybody can see the syllabus? Okay.
Yep.
So this is me. You guys know where my office is. These are my office hours. I will be in my office during these times. So I'm in the office Thursday morning before lab, but then I kill office hours a little bit early so that I can make sure that lab is prepped and things like that. So if you want to see me on Thursday and it's after 9:45, chances are I'm up in White Science 219, making sure that everything is squared away up there. If you want to not come to my office because of COVID, just let me know. We can meet on Zoom. If you need to meet outside of these times, let me know. And once again, I can meet you either in my office or on Zoom, either way. If you want to meet on Zoom, you need to give me time to get a Zoom meeting set up and everything else. And because I'm also trying to kind of limit the time that I'm on campus, once again, just because of COVID, if you just drop by my office outside of office hours, you might not be able to find me in my office. So I think maybe you guys got used to this last semester because of doing a lot of online teaching. The way we do office hours just has to be a little more intentional than maybe we did it before. Don't assume that if you just drop by that you're going to find somebody in their office outside of office hours. Yada, yada, yada. This is what herpetology is about. You can read that yourself. So these learning outcomes kind of reflect the things that we've already talked about. I want you to be able to identify species. That's something you should definitely learn in a course like herpetology. We're going to talk about the distribution of amphibians and reptiles and how that relates to the tectonic history of the Earth. We're going to look at all kinds of things about the biology of amphibians and reptiles. And so knowing those things and seeing how different groups of organisms within amphibians and reptiles differ and are similar in terms of their physiology, their reproductive biology, morphology, et cetera. Global amphibian declines are an issue, and reptile declines are becoming more of an issue. These two things are mainly due to diseases that are going through natural populations of amphibians and reptiles. There is a chytrid fungus that has really impacted amphibians to the point that now you need a permit in the United States to transport amphibians across state lines because they're trying to control the spread of chytrid fungus. And there's also a fungal disease that is affecting reptiles that is especially causing problems in snake populations. And so global amphibian and reptile declines are being driven by a combination of diseases and things like global warming that are just making life a little more tenuous for amphibian and reptile populations than there were 50 years ago. We're going to talk about phylogeny and how we develop phylogenies to recreate the evolutionary history of a group of organisms. And we'll be talking about that relatively soon. And then one of the other things that you will do is you'll create a dichotomous key. So by the time we get out of this class, you guys should be able to have mastered all of these things. This is a hybrid synchronous course. It's hybrid synchronous primarily because the lecture is online, but the lab is going to necessarily be in person. And I don't know why this is actually the statement from my applied stats class, which I need to fix because we're not doing any live coding in here. I guess I was copying and pasting and then forgot to change some of the text. So lab is in person, lecture is online, unless something fundamentally changes about COVID in the next couple of months, which I doubt is going to be happening. The Missouri rollout of vaccines puts me and you, I think, in the last category of people to get the vaccine, because while I'm an educator, I'm not a K through 12 educator. And so I'm just in the everybody else category for vaccine availability. So I think we are all in the same boat there. So we're just going to have to deal with that. And that means that we're probably going to be socially distancing as much as we can throughout the semester. So you need the textbook, which is Poe et al., Harvey Poe. He's a well-established, now he's pretty old, herpetologist. There are only a couple of herpetology textbooks out there. This, I think, is among the more current and more comprehensive in terms of its coverage, which is why I chose it. I just got an email the other day from the publisher. Why did you choose the book? I'm like, because I like it and I think it's good. So anyway, that's what we'll be working out of. Most of the figures and things like this that I'll be using in PowerPoints are from the textbook. I'll also be pulling things in from other places. But there will be assigned readings in here. For example, right now, one of the things that you should be doing is you should be reading chapter 1, which is Why Study Herpetology. And in preparation for next week, you should start reading chapter 2 as soon as you can, because we'll be talking about phylogenetics and how that works. When's the last time you thought about phylogeny and how phylogenies get generated?
Biodiversity.
Okay, I thought so. Biological diversity. So that stuff is probably pretty rusty. So we're going to actually go through that in some detail. The other textbook that you need is the Field Guide to the Reptiles and Amphibians of Eastern and Central North America. This is a book that used to be published by Roger Conant and Joe Collins. Those guys are both dead now. And so Bob Powell, who is actually local here down at Avila University in Kansas City, has taken over the revisions of that. So now it's Powell, Conant, and Collins. This is the most recent update of this field guide. I have numerous iterations of this field guide, but even some of the taxonomy in that current iteration has obviously is out of date. And so you'll find the species still in the book, but once again, refer to the list of amphibians and reptiles in Missouri that I've given you in lab for the actual correct genus and species names for the species we'll be looking at in lab. Then the last thing that you need is some sort of field notebook. It doesn't have to be anything special. It's just something to take notes on while you're in the field. so that as you take photos of organisms and collect organisms for your collection, you have good information on where you found those individuals, what the weather conditions were, what the locality was, what the animal was doing when you found it, et cetera, et cetera. So you need to have some form of field notebook. And I usually take field notes in either a pen that marks in indelible ink or a pencil. Attendance, you guys just know I expect you to be here. If you have accommodations that are Americans with Disabilities Act accommodations, obviously let me know about those sooner rather than later. You guys know about academic misconduct. The most common form of academic misconduct that I see in biology classes is plagiarism. You guys have been trained on plagiarism probably multiple times now, so I really don't expect to see that in this class. I don't see cheating on tests very often, but this past semester in evolution and ecology, people cheated on tests. So once again, I don't really expect that behavior in you guys because you guys know how things work. You're not first year E&E students, and so behave. I'm glad that I know all of you and I've had you guys all in classes before because connecting over the internet is a little more difficult. One of the advantages that we have of meeting together in person in lab is that we have that kind of contact, but do your best to be here, be participatory so that we feel like we're actually having class rather than doing something online, which is actually what we are doing. The one thing I do want to point out here is in order to post the videos online and have them publicly viewed, I need permission from everybody in the class to do that. So if you could drop me an email telling me whether or not you consent to having the video posted on YouTube. If everybody agrees to it, I'll put them up on YouTube and then you can go back and revisit them. And then Zach, who also can't be in class with us because he has another class that there was scheduling conflict with, those will be available to him. Also, I can set up a private viewing system on YouTube. It's just a little more trouble to do it that way. So drop me an email saying yay or nay to making those available. If any of you says no, then I'll come up with a private viewing section. I can't imagine a lot of controversial topics coming up in this class. So anyway, that's that. We won't be having any breakout sessions or anything like that because there's just 5 of you and it would be stupid to break 5 of you up into a breakout session. But there will be times when we'll be talking about papers that we're reading and things like this. And so during those times, I expect you to be as participatory in primary literature discussions as you would if we were sitting in the same room with one another. COVID-19 is a thing, still a thing. So do everything you can to prevent the spread of COVID. Mask when you're around people. Don't be around people as much as you can, et cetera, et cetera. We're over 400,000 people dead in the United States, and that's largely because of our own stupidity. So if we can do what we can. Kelsey, did I just see your eyes go up? You're like, whoa, he just said we were stupid.
I mean, you're not wrong. I just thought it was
in my applied statistics class, as an introduction to why they should care about applied statistics, we recreated an analysis done by the Center for Disease Control, where we looked at. I can actually just pull it up for you. It's a useful and informative paper. Where did I put it? I think it's actually still in my downloads. So this is can you guys see a PDF on my screen?
Yep.
So this is a situation. This is a paper that came out in the Morbidity and Mortality Weekly Report. Yay! COVID has driven me to reading the Morbidity and Mortality Weekly Report at the CDC. In Kansas, there was a mask mandate that went into effect back in July. It went into effect on July 3rd. And the legislature in Kansas, because they are so wise, basically passed a law that said that if a county didn't want to adhere to the statewide mask mandate, they could opt out. That's smart. And that was sarcasm, by the way. And so it set up a nice natural experiment because 24 of the counties in Kansas adhered to the statewide mask mandate, and 81 counties in Kansas refused to adhere to the mask mandate. So it kind of set up this natural experiment. And so what they did was they went and analyzed data on new cases. And I'll just go to the take-home point. So the dotted line represents July 3rd, and the light blue dots are counties that are following the mask mandate, and the dark blue is the counties that did not follow the mask mandate. And you can see that there are more cases per 100,000 individuals, but that's largely because most of those 24 counties that adhere to the mask mandate were urban counties. And so you might expect higher levels of community spread in urban environments rather than rural environments. But what's interesting is that before the mask mandate was put in place, both sets of counties, both types of counties were experiencing increases. But then after the mask mandate, those counties that adopted the mask mandate declined in the rise in the slope of the number of cases. They were going down over time, whereas the slope of the counties that did not abide by the mask mandate had continuing increases in new cases, and kind of at the same rate that they were before the mask mandate went into place. So these kinds of natural experiments are useful in saying, okay, well, we'll do mask mandates work. That doesn't mean that everybody in all of these counties that adopted the mask mandate were actually adhering to masking rules and things like that, but enough of them were that they actually flattened the curve and begin to see a decrease in the number of new cases. So abide by science. You guys are all scientists, do the things that scientists are telling you should do, and we'll hopefully get through this. And after you all graduate, we'll get back to school like normal. This is not exactly the senior year you were hoping for, was it? No. Okay. My apologies for that, but once again, I myself, I see my partner on a regular basis, and I see my chickens. And those are the people I interact with. Where did my PowerPoint go? Oh, okay. Oh, not PowerPoint. I want syllabus back. So anyway, if you are diagnosed with COVID, I'll get a month notification for it, just like what happened last semester. Those of you, how many of you are involved in athletics? Yeah, you guys are in contact with people in a way that other people on campus are not in contact with other people. So you guys especially take care when you're in those groups, because it was unsurprising to me to see that among the groups that had to go into quarantine last fall, among the first groups, actually the first group of a big block of students that had to go into quarantine was actually the football team. And the football team meets every Wednesday for team meeting in White Science. And I remember passing them repeatedly as I was going to the bathroom in the afternoon, seeing them sitting out in the sitting area outside my office in White Science, and 2 or 3 of them were not wearing masks. And so it wasn't any surprise to me at all that COVID began to spread through the football team, because once again, they were openly defying the school's requirement to adhere to masking behaviors. So, just take care, and we'll get through this. I haven't put up the daily schedule for the class yet because I haven't actually finished putting it together. I hope to get that done over the weekend. But on the daily schedule, it'll be an Excel file like I normally do for classes. It'll be posted on Moodle. But on that daily schedule, I've actually highlighted these dates that are the spring break days, which is days that when they're on Monday, Wednesday, Friday, we will not have lecture. And on the 18th of March, we will not have lab that day. Unfortunately, I don't think it's actually going to feel like getting a spring break, but I will not expect anything of you on those days, and you should not expect anything of me on those days. I'm going to treat those days as if I'm actually on spring break. Partly because the provost has said we need to treat those days as actual days off, and partly because I kind of want my spring break. And I'm sure you do too. There's also going to be one day around the middle here, this week of March 16th and 18th. Zach and I need to go and do field work that requires travel to Texas. And so the 19th, that Friday, we're not going to have class so that we have travel time to go and do some field collecting that requires like a 10-hour drive in order to get to the field site. So in that way, at least from this class, you're going to get a little bit of a spring break, but for Zach and I, it's actually just going to be work. So what are you going to do in here? We're going to read some literature, and you're going to write literature summaries. The literature summaries are going to be graded kind of the way I graded literature summaries in E&E. This is something that you should not be surprised about because I'm a one-trick pony. I want you to be able to read and summarize science well, and I want you to be able to critically think about the science that you're reading. And the kind of critical thinking that I'm looking for is critical thinking that thinks beyond the paper that you've read to whatever should be the next steps for investigating a particular set of ideas. I am not interested in the readability of the paper, whether or not you liked it. I'm interested in your assessment of the science and what you think the next steps would be if you were going to be doing work in this realm, whatever this realm happens to be. You will write a lab report. So I talked about this some in lab yesterday, back in April, and I should have started earlier. I should have started in January, but it didn't occur to me until later. As I was bicycling around, I don't know if you guys know, but I bicycle a lot. Are you guys aware of that?
Yeah.
Yeah, I did almost 4,000 miles last year. And as I was biking around in March and April after we had the quarantine, which wasn't really a quarantine, I noticed as I was biking that there was a lot less traffic. Just traffic is always something that you have to be dealing with when you're bicycling. And it's like, oh, wow, it's really nice to bicycle now because there are fewer cars on the road. And so I began thinking about this. And there have been a number of studies looking at roadkill, especially roadkill and the impacts of roadkill on amphibians and reptiles. And I thought to myself, I wonder if the incidences of roadkill are actually decreasing because of the reduced travel, reduced economic activity, et cetera, that is associated with the pandemic. So I roped my partner into helping me with this. And so since April 5th of last year, every other week on Sunday mornings, we've been road cruising the road from 291 to Richmond on 210, and just stopping and identifying every dead animal that we see on the road as we go from west to east. And she has been amazingly patient with me by waking up early on Sunday morning and doing this. We go early on Sunday mornings because it's the least trafficy day of the week, and most of the traffic is like church traffic, and so it's just a little safer. And we do it every 2 weeks. And when we do it, we drag the dead animals off of the road so that we don't count them the next time that we do the road cruising. And then 2 weeks gives us enough time to get some more dead animals on the road. And so what I'm going to do is I'm going to press you guys into service on this project and do road cruising with you guys on Sundays. Because I don't live with you, we will be masked the whole time because we will be in a small car. And what we do is we find each road-killed animal that we encounter. We record the latitude and longitude and the species. We record whether or not it was killed in the driving lanes or whether it was killed in the shoulder, et cetera, et cetera. And that all is going into this big data set. And hopefully what we'll have by the end of the semester, because your data collection will overlap the month of April, which is when we started it last year, we'll be able to compare last April's data to this April's data, as well as just kind of looking at the yearly pattern of roadkill, but also because we're taking geographic coordinates for every dead animal that we see, we can map where animals are getting killed. And I suspect that what we're going to see is we're going to see a spatial patterning to where animals are found. And so there are certain places along this 26-mile stretch of road where we tend to see a lot more animals being killed, and then there are other sections where we don't really see very many animals getting killed. And this probably has to do with habitat structures. So this lab report will be an interesting lab report from my perspective, because it'll be a lab report that kind of addresses, you know, as traveling declines because economic activity is declining because of this pandemic, what does that mean for the incidence of roadkill? But then also this spatial pattern of, you know, are there hot spots along this 26-mile stretch of road where you see a lot of roadkill, and then other cold spots where you don't see as much roadkill? And I think both of those things are going to be borne out by the data. So one of the things that we're going to do in lab over the course of the semester, so we're going to be working on identification, but I think what I'm also going to start doing, probably starting not next week, but the week after that, is every week we'll read a paper to get you ready, get you reading the literature that would go into an introduction for this lab report about roadkill. We'll spend the first 45 minutes or so of lab discussing that paper, and then we'll spend the rest of the time working on identification. And then by the time we've gone through that for 6 or 7 weeks, you'll start to build up a nice literature review for going into the introduction. Does that make sense? Okay. Lab practicals, we'll have 2 lab practicals. We already talked about these yesterday. One will deal with amphibians. The other one will deal with the non-avian reptiles. Identification, classification, geography, etc. And then the personal collection, at least 2 of your specimens must be preserved. The rest can be photographs, but once again, they all have to be documented appropriately. And then the last thing is the dichotomous key. And once again, I told you that this year we're going to divide that dichotomous key up into 2 assignments. Dichotomous key for the amphibians that is due after we've done the amphibians in class, and then a dichotomous key for the reptiles after we finish the reptiles in lab. And the last day to drop is the 16th of April. And so we're going to have 4 exams, 4 literature summaries, 2 lab practicals, a lab report, a dichotomous key. Each half of the dichotomous key will be 50 points. Your herptile collection will be 100 points. That'll give you 1,200 points total. Whatever percentage of those 1,200 points you earn, that's what you earn. There's really no reason, given the students that are in this class, because I know all of you, there's no reason that all of you shouldn't be walking away from this class with A's. So that's what I fully expect, and I would be surprised if I found anything other than that, because I have confidence in you because of my prior experience with you. Grading timelines. I'll probably beat most of these grading timelines because there's only 6 of you in the class, but it takes time to read stuff. And so just this is an expectation for when you can expect things to be handed back to you. This is the grade breakdown. And once again, the course schedule is going to be on the Moodle page, and it'll have all those days when we're not meeting highlighted for you. Any questions about the syllabus? All right. Well, there it is, and it's like I said, it's on the Moodle site. So if you ever need to refer back to it, and the other thing is, if you have any questions about anything as we go along, just obviously, ask. Okay, history. This is not something that is in your textbook, unfortunately. So this stems largely from kind of my own personal study of history as I've kind of grown up being exposed to herpetology by the people that taught me herpetology. And so one of the things that I want to impress upon you, and I should have added this to the end, and I wonder if I have this book in my office or if it's at home. I think it's at home. Yeah. There's a little book that I have called Toads Are Nice People. It's written by a guy named Ron Altig, who is a herpetologist at the Smithsonian. He was a friend of one of my professors at the University of Texas at Arlington. And it basically is a little book, kind of self-published book. Oh, wait, no, I have it here. I just saw it. Toads Are Nice People by Ron Altig. It's actually signed by Ron Altig. It's one of my many science books that are signed by the author. So Butch Brodie, the elder, was friends with Ron Altig. They were in graduate school together. And when they were in graduate school, they went on this cross-country field trip where they basically, they were, I guess were they at Clemson? Where were they? Well, anyway, they're somewhere in the southeastern United States. And they basically drove all around the country, like this big loop around the United States. And their itinerary was basically driven by what are the cool amphibians and reptiles that we have never collected before. And they planned their trip to track those down and find them and collect them and take pictures of them and document them. And the book is largely, it's not that big of a book, was largely a recounting of that trip. And it's interesting reading. And when I interacted with Butch Brodie when he was my professor in a number of my classes at the University of Texas at Arlington, he told some of the stories that when I picked up this book, I saw those stories appearing again in the book. And what's interesting to me about the book is there is a chapter in the book called Chapter 5, and unfortunately the book is falling apart, so I can just take this sheet out. Chapter 5 is entitled Societies, Journals, Men, and Institutions. So that's actually the title of the chapter. I don't know if you guys can see that. But in this, it talks about herpetology kind of as a field. And it's interesting because it says societies, journals, men, and institutions, because herpetology has been a field that has largely been dominated by men, especially at the time that this was published, which was in, there is no date of publication, 1976. So there's a little bit of history in this chapter, but one of the interesting things in this chapter is a thing here at the end. And what this is it's a phylogenetic tree of herpetologists. And so on this, I have a scan of this somewhere, and I'll post this so you can get a little better view of it. But my academic lineage is in this table because Brodie is in this table. And so I can see who Brodie learned herpetology from, and I can tell who Brodie taught herpetology from. So my major professor is a guy named Dan Fermanowitz, and while he worked mainly on spiders and scorpions, he worked on tadpoles as a doctoral student. And his doctoral advisor was a guy named Bob Yeager, who was at Southwest Louisiana University. But his master's professor was Butch Brodie. And then when Butch came to UT Arlington, they ended up hiring Fermanowitz, And Brody was taught by a guy named Bob Storm, and you can tell things about people by who trained them. And so it's easy for me to see in Daniel Furmanowitz the way he approached ecology, the way he approached fieldwork, the way he approached asking questions in science was definitely influenced by Bob Yeager and Butch Brody. And the way Butch Brody approached behavioral ecology was influenced by the people that he studied under. And so oftentimes you can understand the way a person thinks about something by knowing what their academic lineage is. And it's something that I kind of knew, but before I read this book and saw this last chapter, it's something that wasn't really distilled for me until I saw the way Ron Altig was thinking about it. But it's like, oh yeah, that's absolutely true. And so to think that, we want to think about science as this objective search for truth and meaning in life, et cetera, et cetera. But the fact of the matter is that the conduct of science, whether it's herpetology or any other kind of science, is influenced by scientists. And scientists make all kinds of choices in terms of the kinds of things that they study, the reasons that they're studying those things, the approaches that they take to study those things, et cetera, et cetera. Science is far from the purely objective uncovering of facts that we sometimes teach you. It's embedded with all of the biases that humans just naturally have. And some of those biases are actually biases that come from our own training. And so having a historical context for the science that we study is actually useful and informative. And so that's the reason that I try and get history into the classes that I teach. It's important, and we don't talk about it necessarily in this way, but if you read about the history of the development of evolutionary thought, it's important to recognize that Darwin and Wallace, because they were Englishmen in Victorian England, they were kind of, the stage was kind of set for them to think about evolutionary biology in the way they thought about evolutionary biology, because if you read their writings, they view evolution as driving organisms forward in a progressive sort of way. Organisms are getting better over evolutionary time because of the force of evolution. They're becoming better adapted. They're becoming better at doing what they are doing. And this is exactly the kind of thing that when people were thinking about humans in Victorian England, there was this drive that humans should be bettering themselves over time. And so it's not surprising that the way in which Darwin and Wallace wrote about evolution reflects the social mores of the time, if you go back and read their writings. So there is a culture to science, and we can get insight into that culture by studying the history of science. That's a really long preamble to, we're about to study some history. So questions before we get started? All right, then let's go. Darn it, I hate it when it does that. All of my slideshows I have in presenter mode because I have always presented them in the past. And so I have to change that on all of my presentations that I've had in the past. So we can go back to the very beginning and we can look at Aristotle. Aristotle was the first person who tried to present any sort of formal classification scheme for animals. And he put animals into these broad categories. And so we have four-footed beasts that produce their young alive and have hair, and we have four-footed beasts that lay eggs and have scales. And of course, because Aristotle had never been to New Zealand, because New Zealand had not been discovered by European people at that point, he had no idea that four-footed beasts that produce their young alive have hair. he didn't know about the platypus, which is a mammal that is four-footed and has hair that lays eggs. It's one of the few species of mammals that are egg-laying mammals. And he also had not paid attention to the live birth-giving snakes that do occur in Greece. Vipers occur in Europe that give live birth. And so part of this wonky classification system for separating mammals from other organisms is due to his lack of information about weird animals that only occur in Australia and New Zealand that are mammals that lay eggs. And then he also just wasn't very observant about the natural world around him, in that there were organisms that lived in his part of the world that gave live birth, that had scales and didn't lay eggs. And so Aristotle had obviously a very whacked out view of animal classification, not surprising given the time that Aristotle lived. Pliny, another old scientist, he published this treatise called Naturalis Historiae. It was basically just natural history. Many of the animals, this is a comment by a herpetologist, Going. Many of his animals are fabulous, and many of the tales he tells of real animals are equally fabulous, but his accounts are lively, and where he deals with animals he could study himself contain some accurate information. So once again, this was at a time period. So he tried to list all the animals he either knew or had heard about. And unfortunately, he was not terribly factually accurate, because a lot of the animals that he heard about, he did not have good firsthand information of. And he was evidently prone to telling fabulous stories, which makes for good reading, but doesn't make necessarily for good science. So Going was a herpetologist who was at the National Zoo in Washington. He worked at the Smithsonian. Coleman J. Going, and he produced the first textbook of herpetology, which I have right here. This was the herpetology textbook that I studied when I took herpetology. There's Coleman J. Going and George Zug, and Coleman's wife Olive. And herpetology, when I took herpetology now, bigger format books, and also thicker books, because we know more about herpetology than when I took herpetology as an undergraduate. So this was Going's take on Pliny, which basically is Pliny not terribly useful. So one of the things that Aristotle and Pliny did, though, is that they, through their writings, kind of popularized an interest in natural history. And natural history is just this generalized term for the study of organisms and their lives out in nature. And when I was a young kid, I wanted to grow up and become a natural historian. And when I got to my undergraduate program, my academic advisor says, yeah, we really don't do that anymore. I'm like, okay, well, what's the next best thing? He's like, you probably just want to become an ecologist. And so that's what I did. But their writings, when they were published in manuscript form in the 1400s, popularized this interest in natural history. And one of the things that you see in the 1400s, 1500s, 1600s is this rise in the interest in the lives of organisms. And especially the 1500s into the 1800s was a period of rapid growth in more formal studies of the lives of organisms, both plants and animals. So we get to things like John Ray, who was the first person to group amphibians and reptiles together. And this was really the first real attempt at systematizing the taxonomy of amphibians and reptiles. Carolus Linnaeus, or Carl von Linné, So Linnaeus, you oftentimes see his name written Carolus Linnaeus. This is the Latinized version of a German name. His German name is Karl von Linn. And so because he Latinized all of the animal names, he also Latinized himself. And that's the origin of Carolus Linnaeus. He wrote a series of volumes in the 1750s that came to be what is known as the Systema Naturae. And he's the person who introduced binomial nomenclature, the idea that every organism belongs to a genus, and then within that genus, each species gets its own specific epithet. And I wrote this on the board in lab yesterday, but you might not have gathered it. A species name is made up of a genus name and then a specific epithet. And the specific epithet is sometimes just referred to as the species name, but formally it's called the specific epithet. So Eurycia lucifica, Eurycia is the genus and lucifica is the species, the specific epithet. Linnaeus, needless to say, didn't have a lot of respect for amphibians and reptiles. These foul and loathsome creatures are distinguished by a heart with a single ventricle and a single auricle, doubtful lungs, and a double penis. Most amphibia are abhorrent because of their cold body, pale color, cartilaginous skeleton, filthy skin, fierce aspect, calculating eye, offensive smell, harsh voice, squalid habitation, and terrible venom. And so their creator has not exerted his power to make many of them. Needless to say, he's not a fan of herpetology. So after binomial nomenclature came along, that served as kind of a method for kind of organizing organisms into a classification scheme. And that actually helped science out a lot because we could talk about things and whether or not they shared certain characteristics with one another. And if they shared characteristics with one another, they were put into similar groupings. Those groupings became families and orders, et cetera, et cetera. And we began to develop the taxonomic hierarchy that we talked about in lab yesterday. So by the 19th century, so remember that the 19th century is the 1800s, we began to start getting people in museums. So with the rise of interest in natural history and the rise of global exploration by European superpowers. So remember at this particular time in history, the big superpowers in the world were England and France and Spain and Portugal and Italy. And these nations all had navies, and these navies went out and basically established colonies, established territories that are part of those colonial powers. And in these new areas where Europeans had never been before, there were all of these crazy new species that nobody in Europe had ever seen. And so with the rise of interest in natural history, coming at the same time that we had navies that were going out and discovering continents that were previously unexplored and exploring those, you had this huge upswelling of interest in the organisms that were coming from these strange and exotic places. These specimens were deposited in museums that were formed to hold these specimens in Europe. And so some of the oldest natural history museums are in Europe, and they have very large collections that go back literally centuries. There were people who needed to classify and name these organisms as they were being brought to the museums and then curating them while they were there. And so you begin to see a rise in a professional class of ornithologists, botanists, herpetologists, mammalogists that were in charge of these collections. And so a lot of these people early on in the history are curators at these big national museums. So we have France in the 19th century. Dumeril and Biberon produced a thing called Herpetologie Générale, basically general herpetology, which was once again 10 volumes, once again an attempt to kind of catalog all of the specimens that were in museums in Europe at that time. Dumeril's boa is named after Dumeril. And so oftentimes in the taxonomy of herpetology and other organisms, you oftentimes see species being named after people. And so if you look at your list of herps from Missouri that we were looking at yesterday, you see under frogs, you see Staphyopus holbrooki. Holbrook was an early American herpetologist that did a lot of herpetology on the new acquisitions to the United States of the West. Anaxyrus fowleri is named after a herpetologist. Woodhousei is named after a herpetologist. Blanchardii is named after a herpetologist. So the process of naming, giving a species a specific epithet, that epithet is often referred to as an honorific if it is named after a particular individual. And in some cases, genuses or genera are named after individuals as well. And so Acanthophis dumerili is one of these species that is named after an early herpetologist. Gunther looked at neotropical amphibians and reptiles in, once again, the 1800s. Once again, 9 volumes on the amphibians and reptiles of once again this newly explored area of the world. Boulanger, another French herpetologist, but who was working at the British Museum, also published published a 9-volume set of treatises on the specimens at the British Museum. So in all these cases, people were basically looking at their local museums and then cataloging the specimens and describing the specimens that were at those museums. At the herpetology meetings in the summer, there is an auction that is held every year, and the auction is basically to support graduate student travel to meetings and graduate student research grants. And every 5 years or so, Boulanger's volumes will come up for auction, and they always get sold for an insane amount of money because it was one of the first really good treatments of herps at the time. And so they're kind of collector's pieces now. Holbrook, which I just mentioned because we have Scapiosis holbrooki, he was one of the first North American herpetologists, and he was around at a time when the United States was expanding into what became known as the American West. And so he found all kinds of crazy organisms. In some cases, organisms like Gopherus polyphemus, which is the gopher tortoise in the southeastern United States. You find them commonly in Florida. Crotalus adamantius, which you saw photos of in the introduction that I did on Wednesday. The eastern diamondback rattlesnake, once again limited to the southeastern United States. But then because he was studying specimens that were being found as naturalists went out into the American West, he also described Phrynosoma cornutum, the Texas horn lizard, which is only found west of the Mississippi. And so he wrote a big fat book called North American Herpetology that once again attempted to describe all of the species that were known from collections that were done as the United States expanded into the American West in the early 1800s. When Lewis and Clark went through and explored the drainages of the Missouri River and the Columbia River, they collected a lot of scientific specimens and brought those back, and those were put in the Smithsonian for the most part, and because that was a government-funded expedition, and many of those specimens were the kinds of things that Holbrook was looking at when he was doing his work. Edward Drinker Cope, we've already encountered him because one of the gray tree frogs is named after Cope, Cope's gray tree frog. He was quite a character. Once again, he went with the U.S. Geological Service during these expeditions that were exploring the American West during westward expansion of the United States. He did a lot of work on amphibians and reptiles in general, but he was also involved in what became known as the Bone Wars with a guy named Othniel Marsh. There have been books written about these two guys because in addition to working on extant amphibians and reptiles, both Marsh and Cope worked on fossil amphibians and reptiles, more particularly fossil reptiles. And so they would stake out claims on these fossil deposits in the American West that were well known for containing dinosaur bones. And they would fight furiously over access to these sites. They would keep these sites safe, very secretive. And if one or the other found out about them, they would actually go to great lengths to keep the other individual from gaining access to their fossil beds. Cope is also an individual that has a journal named after him. So one of the herpetology journals is called Copia. I think I have a copy of it in my office. Copia, scientific journal devoted to the study of amphibians and reptiles and fishes, and it's named after Edward Drinker Cope. And just recently, actually last year, given kind of the recognition that we honor people who are maybe not very honorable, the American Society of Ichthyologists and Herpetologists, which is who publishes Copia, has changed the name of Copia because, as it turns out, Edward Drinker Cope was not a very nice guy in life. He was a big advocate of segregation. He had very low opinions of women, and they decided that we are no longer going to honor Cope by naming a journal after him. So we'll stop there because we're almost out of time, and we'll finish off the 19th century and then get into 20th century herpetology at the beginning of the class on Monday. Questions? All right, have a good weekend, and I'll see you on Monday.